It's quite an honor to present today some of our work on magnetic particle imaging, and in particular, I'm going to focus on hemodynamic imaging in this talk. My name is S.Conolly, and I work I'm a professor at UC Berkeley in both bioengineering as well as EECS, and I'm really excited to present today. So again, I'd like to thank the NIH Brain Initiative for this presentation on magnetic particle hemodynamic imaging. So magnetic particle imaging is a new imaging method to track super-paramagnetic iron oxide nanoparticles as tracers through the body. It has a lot of advantages over current imaging methods, including MRI, which is another method to image SPIOs. It has zero radiation, it has very rapid preparation time, often right from the refrigerator, and it has no half-life limits, especially relative to nuclear medicine techniques. And in fact, MPI actually competes mostly with nuclear medicine, not with MRI. It has remarkable signal-to-noise ratio already on the order of 1 micromolar, and it has almost the best dose-limited sensitivity already. The contrast-to-noise ratio is exceptional because there's zero signal from background tissue and zero attenuation through tissue. And it's great that, unlike MRI, MPI is perfectly robust to normal magnetic field tolerances inside the body. 5% field tolerance is totally fine. So we have no trouble whatsoever imaging in the lungs, unlike MRI and ultrasound. And this could soon be an ideal angiographic modality. And so that's what I'm going to focus on today. So this shows a picture of the nanoparticles. They're about 25 nanometer in size. We had to build our own MPI scanner at UC Berkeley. We actually spent about 6 years building 3 different MPI scanners. And this is what it's all about. This is the application that makes sense of it. So we're competing here with red blood cell scintigraphy to see a GI bleed, a gastrointestinal bleed in this animal. And you can see this red hue is the leakage of or extravasation of the nanoparticles out of this GI bleed. And you can see in the control animal, absolutely no extravasation. This is just an anatomic reference X-ray scan. So I'll try to explain how MPI works to create this image today. So again, MPI sees these SPIO particles, but it's with positive contrast. Very often with MRI, you actually see it as a so-called T2 star dropout in an image. Instead, all we see in MPI are the nanoparticles, and they have incredible sensitivity because they're so intensely magnetic. So we can actually image either blood vessels, as shown here in this phantom shot, or we can actually track molecular tags, and I'm going to talk about that in a different talk. So there's zero background signal, so you have great contrast to noise ratio. It's linear in positive contrast, perfectly linear in the number of nanoparticles per voxel. We can currently see 200 cells anywhere inside of an animal, and we think we can get this down to maybe fewer than 10 cells. And it works at any depth. Again, it has an infinite half-life, unlike hyperpolarized C13 methods. There's no loss of magnetization with time. They remain super-paramagnetic forever. And so we've been able to image stem cells after 87 days inside the animal. These images illustrate how MPI can see through tissue. This is real data. We did a 3D projection reconstruction image set and then displayed it as a MIP. And you can see that there's no difference whether we surround the helical phantom with tissue. So how does this work? We actually exploit the nonlinear Langevin physics of SPIOs. And unlike MRI, where you're always saturated, in MPI, we're actually moving from negative to positive by changing the magnetic field through this 5 millitesla saturation field. And you can see that the saturation in each particle is very intense, 0.6 Tesla. And this electronic magnetization is actually 22 million times stronger than the equivalent nuclear paramagnetism that we image in MRI. And so that's why, in a nutshell, why we can actually image with micromolar sensitivity. And again, don't forget that we can image this forever. All right, so this is how we actually make an image. So if we have a really strong magnetic field gradient, and unlike MRI where we just flip the amplitude, but we keep the origin still, in MPI we slide the origin of the magnetic field gradient all the way across the animal. So let's say you had SPIOs in the left side of the brain and the right side of the brain. They're both going to be pointing down at the beginning of the scan. And as we slide this gradient field over, the spins on the left one are going to first flip, and then the ones on the right are also going to flip. And then we're going to induce an EMF in our receiver coil shown here. And so what this makes clear is time maps directly to position. And that's actually the basis of the X-based theory of MPI that we developed 10 years ago, published here. And so we actually showed formally that the point spread function of MPI is actually proportional to the derivative of this Langevin curve. And so what this means, the sort of bottom line in terms of resolution, today's particles give us about 10 millitesla magnetic resolution. And then the strongest magnetic field gradient that we can make for a small animal, really strong, about 7 Tesla per meter, much stronger than MRI, we're getting about millimeter resolution. But I'm going to talk very soon about something that can give us an order of magnitude improvement, because this is the only weak attribute of MPI today. So today I'm going to talk about hemodynamic applications, including traumatic brain injury and stroke, lung ventilation perfusion, and also the GI gut bleed that I talked about earlier. And these are the collaborators I'm working with. So why, what is MPI's superpower? I think it's really about capillary level perfusion. The spatial resolution is still not the strongest point, but because we have no signal in the background, MPI doesn't suffer from so-called partial voluming effects, where the background sort of bleeds into your image. And so you can actually make an image where shows you the blush due to capillary level perfusion in this small animal. And so as a result, we've been able to publish some of the first in animal traumatic brain injuries. And here you can actually see the perfusion defect after a traumatic brain injury on the right. We've also been able to make very preliminary CBV and CBF, which could be useful for both stroke as well as functional MPI. And so this has been sort of the excitement behind our Brain Initiative grant. And again, the fact that we have capillary CBV can actually help a great deal getting a nice smooth CBF map, and there's no background signal to cause that partial voluming in MPI. One of our first preclinical applications was actually pulmonary embolism detection, which is a very big application with more than 500,000 pulmonary embolisms and deep vein thrombosis each year. This is typically diagnosed with CT pulmonary angiography, but for patients with who have chronic kidney disease or pregnant women, they prefer to use the more venerable study of ventilation perfusion. And so MPI lines up beautifully to replace nuclear or to form a radiation-free complement to the traditional ventilation perfusion study. The big problem with this is it's about 3 to 5 hours, as well as having radiation. And so the way we did this was to traditional perfusion study, we used 40 micron macroaggregated albumin labeled with the nuclear tracer, technetium-99m. And this, of course, traps in the 6 micron capillaries of the lung. And so you can get a nice appreciation of the lung. So we were able to just replace the technetium reporter with an iron oxide reporter. And then instead of a 3-hour or 1-hour study, we're able to go all the way to a 9-minute study. And you can inject this directly from the refrigerator. You can't do that with this one. You have to actually do the MAA hot chemistry just in time. So this could be really practical for especially pregnant women and chronic kidney disease patients. We can also do the lung ventilation study, as demonstrated in this paper a couple of years ago. And again, no hot chemistry is probably the most compelling piece for radiologists, but it's also great that there's no radiation. And you can sort of see here the ventilation part of the study. Another application we've worked on with the UCSF surgeon, John Carter, GI bleeds, I've already talked about, they're very common, and often RBC technetium-99 scintigraphy is used because it can detect the smallest bleed. It has some drawbacks, including 4 millimeter spatial resolution and a pretty slow labeling and scan time together about 3 hours. And so we can actually replace this long hot chemistry lead time, and you can actually just inject directly from the refrigerator again. And so I've already shown this data set here. Here, all we used was a long circulating tracer. It's got a 4-hour circulation half-life due to PEG coating. This was developed by a startup company in Washington called Loadspin Labs. And so you can actually see this extravasation here. And remarkably, we actually beat the dose-limited sensitivity of the scintigraphy study, which is quite amazing. This is the time course of the study over a couple hours. You can see the gut bleed model. What we did is a digital subtraction angiogram to remove all of the circulating particles from the scan. And then we can see the particles that are actually extravasating from the gut bleed over this period of time. And here in the control animals, you can see how steady that digital subtraction angiogram worked. And you can read about this in ACS Nano just a few years ago. Probably the most exciting thing we're working on right now in the lab is trying to improve what I would call the only remaining significant weakness of MPI, which is its spatial resolution. So we're doing some work with Professor Carlos Rinaldi at University of Florida, and we were able to produce this image pair in vitro on the right. And I want to stress there's no deconvolution here. This is just a much higher spatial resolution particle or magnetic resolution particle. And remarkably, we get not only a resolution boost, we also get an SNR boost. And so I wanted to really understand this. The last 2 years, my lab has really been devoted to figuring out how these work and how we can get them into an animal. So we've developed a chaining hypothesis that what's going on is these SPIOs are actually forming a magnetic chain, and that each dipole field from the adjacent particle is actually stronger than the field around it. And you're getting a coherent effect, sort of an amplification, if you will, due to positive feedback from being in the chain. And this makes sense because the applied field that we normally flip at is only around 6 millitesla, whereas the SPIO field, the dipole field emanating from that is at least 1 order of magnitude, maybe 2, stronger. And so there's already in the literature, a name for this is called super-paramagnetism. And so just to show that we think we're on the right track here in this chaining hypothesis is that, you know, the data that we show here in our one-dimensional MPI scanner for super-paramagnetic particles, you see this cliff in the magnetization versus H curve, the MH curve, whereas super-paramagnetic particles are relatively shallow. And this is what gives us both the huge signal-to-noise ratio per picogram, as well as a really sharp point spread function. Now, it also gives us some headaches because now we have coercivity that we need to deal with and remnants, but we're willing to pay the work around that with more advanced pulse sequences. So to show that we actually think we have a handle on this, you can see that our simulation of the dipole field included in the Langevin physics actually recapitulates our data beautifully. And so we're routinely now with particles, you know, developed in my own lab, we're routinely getting 1 millitesla resolution, which is that order of magnitude we were going for. And in this data here, you can actually see both a 16-fold better resolution and signal-to-noise ratio. But it was crucial that we took care to make sure that we treated that coercive threshold properly. All right, so in conclusion, I wanted to stress, this is a very fast presentation, but I want to make sure you realize that MPI has extraordinary sensitivity, already 1 micromolar, and with these new super-paramagnetic particles, maybe even 100 nanomolar, we have unbelievable contrast and safety already. MPI already offers advantages remarkably over scintigraphy and SPECT. I wouldn't claim that we're already competitive with PET in terms of cancer tracing. We think we can improve MPI resolution by up to maybe even more than 10-fold using these super-paramagnetic particles, but we haven't got it yet working in vivo, and so we're working on that right now in my lab. And this is quite amazing because we can actually reduce the cost of MPI more than 100-fold if we can actually reduce the gradient by a factor of 10. So I want to acknowledge all the funding from the NIH and these other sources, and I would like to ask any questions.